I was wondering about, I know that ALS is considered essentially untreatable. I mean, I know that they do have some drugs that slow it down or maybe allegedly help, but I was just wondering if functional medicine has shown any evidence of slowing down the symptoms of ALS. We recently, kind of a friend of a friend was diagnosed with ALS, so that's why it was on my mind.
Okay. Well, good question. And for those who don't know, ALS is amyotrophic lateral sclerosis, or otherwise known as Lou Gehrig's disease. So yes, it is considered a progressive neurodegenerative disease without cure. And even cause isn't fully figured out yet, but there's each day science is figuring out more. So we'll dive into that today. And so, you know, there's no, just to lead off, there's no say functional medicine magic, so to speak, that can stop it. But if we can understand the mechanisms of the disease, then we should be able to figure out clinical targets. So that's what I want to walk you through today is using the research. Well, what do we understand about the disease right now? And is there anything actionable from what we understand about the mechanisms of the disease? So we'll start with this study from Frontiers in Cellular Neuroscience from 2015. And the title is ALS Patient Stem Cells for Unveiling Disease Signatures of Motor Neuron Susceptibility, Perspectives on the Deadly Mitochondria, Endoplasmic Reticulum Stress, and Calcium Triad. So it's a mouthful, but basically this study is trying to figure out cellular contributions to the disease process. And so what they do is they dive in. I'm going to read to you a little bit just so everyone's on the same page about the disease process, and we'll go from there. So ALS, or Lou Gehrig's disease, is one of the most common neurodegenerative disorders and adult onset motor neuron diseases. The disease is primarily characterized by a relentless degeneration of upper motor neurons in the cerebral cortex, which is up in the brain, and lower motor neurons in the brainstem and spinal cord. Symptoms are muscle weakness, progressive paralysis, speech and swallowing disabilities, and ultimately leads to death by respiratory failure. And again, it's once the neurodegeneration and the upper motor neuron lesion and lower motor neuron lesion, once the lower motor neuron lesion reaches the phrenic nerve and the respiratory centers in the brainstem, that's when it's all over. Currently, ALS is considered a multifactorial syndrome with motor system degeneration only a part of the more widespread disease process. Increasing evidence over recent years emphasize that in addition to motor neurons, other cell types such as astrocytes, microglia, oligodendrocytes, and muscle cells may also actively participate in disease processes such as neuroinflammation and muscle degeneration. So they go on to say that recently there's been a big push to figure out a genetic contribution to this disease. And they talk about how there's a familial, the disease pretty much occurs in two ways. There's a familial side, which would appear more genetic, right? If it's happening more in families, the families likely have genetics in common. And then there's the idiopathic version, which is more or less explainable. So we move forward with these three genes alone account for more than half of the reported familial ALS cases, making RNA dysmetabolism one of the central issues of ALS pathogenesis. So pathogenesis is, how does something happen? Well, they just said dysfunction in how RNA is handled appears to be one of the central issues. So that's something we could say, you know, from a functional medicine perspective, do we have any strategies or any tools in our belt to address RNA dysmetabolism? So we can return to that. Mutations in these genes have been also linked to oxidative stress, okay, which, you know, metaphorically, I've spoken with you guys many times about rust would be the metaphor for oxidative stress. Protein misfolding and aggregation or buildup, endoplasmic reticulum and cytoskeleton alterations, ubiquitin protein pathway malfunctions, blah, blah, blah. Glutamate-mediated excitotoxicity. We've talked about neuroexcitotoxicity and glutamate, so that could be another option for us. Calcium imbalance, another actionable place. And axonal transport defects. So these are all potential clinical targets for us because these are mechanisms that appear to be involved in the disease process. And then we hit the big one. Mitochondria play a key role in cellular respiration by converting nutrients into ATP, and ATP allows us, is the energy substrate we use to heal or do whatever we're going to do. Mitochondria are also the main source of reactive oxygen species. So mitochondria are the main source of oxidative stress as well, and act as gatekeepers in intrinsic apoptotic pathways. So apoptotic pathways, that's programmed cell death. So our cells have programmed cell death. They go through so many, say, replications or so many divisions, and then apoptosis is programmed cell death. So they die in an anti-inflammatory way, versus necrotic cell death would be a pro-inflammatory, non-planned death. And that's what we don't want. Mitochondrial dysfunction can lead to oxidative stress, failure of cellular bioenergetics. So think about the pH discussions we've had before, okay? And ultimately to cell death. ALS patient tissue and animal models exhibiting mitochondrial alteration and dysfunction have frequently been found to also exert endoplasmic reticulum stress as a cellular compartment where secreted and membrane proteins are made and folded. So the endoplasmic reticulum is where proteins are made and folded. As the center where this happens, the endoplasmic reticulum is equipped with things that help the proteins fold, okay? When those things like foldases, chaperones, and cofactors to process the proteins are misfolded or aggregate, then we have issues. So you may have heard of Parkinson's disease and Alzheimer's disease, and you may have heard of beta amyloid plaque buildup in Alzheimer's disease and tau protein buildup in Alzheimer's disease, or Lewy bodies in Parkinson's disease. These are protein aggregates that basically you could think of, gunking up the brain and inhibiting optimal function and driving inflammation and then neurodegeneration, while ALS is showing a form of this as well. Stress conditions can interfere with the endoplasmic reticulum function and result in abnormal folding and aggregation of proteins. We're almost done, hang in there. Despite enormous advances in understanding the pathogenic events occurring in ALS, only a small fraction of all reported cases can be linked to genetics. Okay, so that's cool, right? On one hand, it's not cool because we want an explanation. On the other hand, it's cool because we can't change genes. So if only a small fraction of all reported cases are due to genetics, that means a large fraction is likely due to something outside of genetics. And that something, if we can nail it down, is something actionable, right? So thus the cause of the non-familial ALS is largely unknown. And while environmental parameters have been a subject of many studies, unidentified genetic factors are also thought to be contributing. So this is what I don't like, because obviously, if you don't know, most scientific research is funded by pharmaceutical companies, because if you find genes or you find medications that work, pharmaceutical companies can make a lot of money. So to investigate environmental causes that don't result in blockbuster drugs, it's not really high priority. And that's sad, because most disease is environmental or lifestyle or epigenetic, as we've spoken about before, not genetic. So, you always see a sentence like this in there that kind of is like, yeah, it might be environmental, but let's focus on genetics. So that's the obligatory sentence there. But mitochondrial dysfunction and endoplasmic reticulum stress have been implicated as major events in motor neuron degeneration. So that may have been awful to sit through, but I just wanted you to see the science explaining what these pictures are going to show us. So now we have this graphic here, and the graphic is showing us, the title is Oxidative Stress, Protein Misfolding, and Mitochondrial Dysfunction are closely related, okay? So basically when you start when you start in one area here, it's going to cascade to the other areas. What do I mean by that? Okay, so this big long S-shaped thing is a picture of a neuron. Okay, and this pointy spot, a spiky thing over here is called the dendrite. Okay, and down here is the other end. So the long part is the axon, and down here is another end, or the synapse, the neuromuscular junction in this case. So this is a picture of one neuron, and then we're zooming in to see in the dendrite is the cell body where the nucleus is and where all the action's happening. And so if we zoom in there, we can see, all right, mitochondrial dysfunction. Well, if we start at mitochondrial dysfunction, you can see arrows going to endoplasmic reticulum stress, then to oxidative stress to further mitochondrial dysfunction. Or mitochondrial dysfunction might cause oxidative stress first, which causes ER stress, which causes mitochondrial dysfunction. And so you can start the cascade at any point. So if we can intervene at one or more of these areas, then we have, tools to decrease or optimize mitochondrial dysfunction, decrease oxidative stress, optimize protein folding and aggregation. And so maybe we can help support or slow down the disease process and progress. So that's where we would, from a functional medicine perspective, that's where we want to address that, okay? And how we would want to address that. So if we can jump to a different screen here for a minute, I think a different graphic will help us understand further. Where is it? Oh, I don't need to change screens. I've got it right here. Sorry. Boom. All right. So here's something to understand about all neurologic disease, not just ALS. Okay. We're talking about brain and we're talking about neurons. Okay. So down here on the left, you can see healthy neuron resting membrane potential. All right. Right here. And you'll notice that negative 70 millivolts is a healthy resting membrane potential. So at negative 70 millivolts, your neurons are resting. Okay, at negative 40, once you cross this negative 40 threshold, so you raise the millivolts, once it crosses that negative 40 threshold, bang, you have an action potential. So now a neuron fires. And you might have a thought, or you might have a movement, or you have, secretion of a neurotransmitter, some function is happening in the brain and neurologically. You'll see here that key to brain function is ATP, and key to ATP production is healthy mitochondria. Well, key to healthy mitochondria is healthy oxygenation, healthy glucose, healthy stimulation, so exercise, movement, playing instruments, novelty, that's what the brain loves. And then, healthy redox, so reduction and oxidation balance, okay? So if the brain gets what it needs, and the brain needs oxygen, the brain needs glucose, the brain needs stimulation, if that's happening, we have healthy brain mitochondria, healthy ATP production, healthy brains, all right? As we just talked about, unhealthy mitochondria lead to these things and unhealthy brain. And so in the context of this conversation, ALS, well, how could we suffer from unhealthy mitochondria? Yeah, maybe there's some genetics defects that play in there, but there's a lot of lifestyle and environmental things that can drive it. Poor oxygen will do it. How could that happen? Well, if you're anemic for any number of reasons, one of which is iron deficiency, well, then you're not carrying oxygen well to the brain, and brain requires 20% off the top at rest. And when you're more active, it's going to require more. So if we're not carrying oxygen well, that's not good. And then if we're not delivering it well because we have poor circulation to the brain, that's not good. So say you have low blood pressure, right? What's one symptom of low blood pressure? Lightheadedness, dizziness, right? Why? Well, because the brain is an extremity and it's the highest up in gravity. So if we have low blood pressure, it's hardest to get that oxygen up into your head. And so we may not deliver oxygen ideally. And, in the acute short term, that might be dizziness or lightheaded, but chronically over years, that might be poor oxygenation leading to poor mitochondrial function. So if you're chronically anemic, say. And how many times have we talked about me seeing patients that have been anemic for years and their doctors never do anything about it? Okay. Same thing, blood sugar issues. If you have chronic blood sugar issues, you can have unhealthy mitochondria. Well, you know, we all know people who are hangry or who have insulin resistance or diabetes. All of those things are blood sugar issues. So if we're not delivering glucose to the brain, again, it's not going to function optimally. And if it gets real bad, you know, insulin resistance of the brain in some circles is called type 3 diabetes, which is a known driver of Alzheimer's. So blood sugar is key, and Alzheimer's is one neurodegenerative disease that is evidence of that. Lack of stimulation. If you don't exercise, if you don't learn new things, if you don't stay in community, that can lead to poor mitochondrial function and poor brains. So what do they tell the elderly? Exercise, okay? Stay in community, go play bridge, go play tennis, you know, learn an instrument, do something new to continue to stimulate that nervous system. And then lastly, anything that's going to drive inflammation and oxidative stress, it can mess up your mitochondria. So autoimmune disease, okay, we'll talk about in a minute how it's undetermined yet if ALS is definitely an autoimmune disease, but there's connections there. Environmental toxins, heavy metals, you know, mercury, lead, aluminum is huge from an excitotoxicity standpoint. Glyphosate, you know, any other head injuries for inflammatory triggers. There's lots of things that can damage mitochondrial function, which then raise resting membrane potential of your neurons and make it easier to fire, okay? And that's not necessarily good, as we'll see in a moment with excitotoxicity. So hopefully this picture is a little bit easier to understand for you than this one, but that's what we're talking about, okay? Now let's jump over to another study. This is microglia and neuroinflammation and ALS and frontotemporal dementia. Okay, so this C9 ORF72, this is the gene that is considered the most commonly mutated gene in ALS. And this study's looking at the connection between neuroinflammation, which is just brain inflammation, and ALS, and frontotemporal dementia, which is similar to ALS and similar to Parkinson's in some instances. So what they say is this gene is the most commonly mutated gene in ALS. It has an important role also in myeloid cells, which are immune cells, and open the possibility that altered microglial function plays an active role in disease. Okay, microglia are the immune cells of the brain. If you alter their function or you activate them, they become pro-inflammatory and they start fires in the brain. Well, then a fire in the brain is neuroinflammation, okay? And neuroinflammation chronically is not a good thing. That drives neurodegeneration, may open you up for autoimmune disease, bad stuff. So in this paper, they're discussing that, and they're also discussing the potential connection of autoimmunity and ALS. So let's go check that out. So I'll just read to you a little bit here about the autoimmune side of things, since you know that's a common topic for us. The idea that autoimmunity is associated with ALS has been appreciated for decades. ALS patients have a high frequency of monoclonal paraproteinemia, which is monoclonal proteins in the blood, and a high incidence of autoimmune disorders. Numerous IgG antibodies and complement proteins are found in blood from ALS patients, and antibody titer was positively associated with disease severity. Monoclonal antibodies are detectable in 60% of sporadic ALS patients, so the non-familial caused or associated ALS patients. And autoantibodies against ganglioside GM1 and GD1A, sulfoglucuronal paragloboside, neurofilament proteins, and TNF receptor family member FAS, and voltage-gated calcium channels have all been reported, indicating that ALS patients generate antibody responses to a variety of autoantigens. Okay, so there appears to be autoimmune processes in up to 60% of non-familial based ALS patients, which fundamentally would implicate the immune system. Okay, So regardless of the target of the antibodies, if we're attacking self, we're losing tolerance, self-tolerance. We're losing the ability to say, that's Scott, don't shoot at that. That's not Scott, shoot at that. Okay, if we're shooting at Scott or we're shooting at self, that's autoimmunity. More recent epidemiological and genetic studies continue to support the association between ALS and autoimmunity. A large study found that there were significantly more cases than expected of ALS associated with a prior diagnosis of asthma, celiac disease, juvenile onset diabetes, which is type 1 diabetes, multiple sclerosis, myasthenia gravis, lupus, ulcerative colitis, supporting that autoimmune disease and ALS might share common genetic or environmental risk factors. The recent identification of a loss of function mutation to TBK1, which is a well-characterized gene regulator of innate immunity as a cause of ALS, further reinforces this concept. It is also possible that instead of autoimmunity directly causing ALS, the two conditions arise from a shared genetic or environmental risk factor and simply co-occur in some individuals and families. So if we look at this graphic here, the take home is that these authors are suggesting a possible, what they're calling a dual effect. Let me get my highlighter. Possible dual effect of the C9orf72 gene repeat, okay? One effect is that it causes a loss of function of microglia with a pro-inflammatory state. So basically it leads to activated microglia, which drives inflammation in the brain. And then it leads to a gain of function in other neurons, leading to RNA and protein aggregate toxicity. What is RNA and protein aggregate toxicity? This guy, what we talked about earlier, okay, the misfolded proteins and aggregates. So this is a newer study, and these authors are saying, hey, this most common genetic variant seen in ALS appears to have a dual effect of activating microglia, activating brain inflammation, and driving protein aggregates, okay, which are two parts of the disease process. So to kind of wrap it up, if we look at autoimmunity and immune dysfunction, inflammation is central role. If we're looking at mitochondrial dysfunction, oxidative stress, protein misfolding, inflammation plays a central role. And we saw in this study, one of the key factors of pathogenicity suspected is excitotoxicity. Where'd it go? It was in there somewhere. We talked about it earlier. So I would point you to this video, which I made years ago. It's called Excitotoxicity, Migraine, Depression, ADHD, Constipation, Memory Loss, on my YouTube channel. And what I do is I walk you through neuroexcitotoxicity here and show you that when excitotoxicity is happening, neurons blow up. They degenerate, drives brain inflammation, and it can drive, migraine to Parkinson's to ALS. Okay, so you can watch that video. And what's happening there is you have too much calcium influx into the cell. And when you have too much of that, you blow up neurons. So to help answer Scott's question, what we would do to help an ALS patient, even though functional medicine doesn't necessarily have the quote unquote answer protocol to ALS, is we would want to address anything we find over here, right? So anything that is on this side of the equation in the person, if we can help with that, then maybe we can slow down the progress of the disease and give them more quality of life for however long they have left. And so we've spoken on a previous video as well about morbidity compression. And morbidity is suffering, mortality is death. And so the idea of morbidity compression is we want to decrease the amount of time in someone's life that they're suffering so that they suffer for as little time as possible in their whole lifespan. So instead of the typical American having quality of life until age 65 and then suffering until, 72, 81, however they make it. We want them to have quality of life until, say, 75, 76, and then die at 81, right? Or ideally, they'd live great up until the end, right? So we want to compress morbidity, and if we can find issues and this side of the equation and address them, then we can help compress morbidity in that person and optimize quality of life. So let's open it up for questions and comments here.
So would one of the first things, if this were a patient of yours, that you would want to do is to find out if they have that mutated gene or not? Is there a test for that?
I'd have to see if there's a clinical test for that. They can test for it. Sometimes what you can test for in the scientific research is not commercially available. So I'd have to look that up. I don't have that answer right now. But if it's a commercially available, we could look at it and depending on price, see whether it's worthwhile, because if it's the most commonly mutated gene in ALS, we might safely just, assume it's there, and because we can't change it, move on.
Work around it.
Yeah.
Yeah.
Right. Because we're not going to change the genes, but if it's the most commonly mutated one and its function is to drive microglia and protein aggregation, then we would work against the microglial activation and the protein aggregation. Make sense, Scott?
Yeah. I was going to say before I told you that ALS is not considered autoimmune, but it's pretty obvious it has some autoimmune components.
Right. Yeah. And interestingly, this study had said that the GM1 antibodies are present, wherever that was. Here it is. Ganglioside GM1 antibodies. I'm going off the top of my head now, so I could be wrong, but I believe those are also positive in Guillain-Barré syndrome. Which is a known autoimmune issue. So again, there can be overlap. Maybe maybe like this paragraph says it's happening, both are happening in the same person. That matters to a point, but if it's there, it is the same person, and we're not addressing ALS only, right? If there's autoimmunity present, we have to address that too, because that is going to impact the ALS and vice versa if it's not driving the ALS.
I just think that chart with the left and right, the healthy and the unhealthy, it just pertains to so much.
Yeah, it goes
so much.
so deep.
Yeah, and like just that everybody that comes to see you probably has something on the right-hand side that, needs addressed.
Yeah,
So,
likely, because they wouldn't come to see me if they didn't usually. Most people aren't proactive, unfortunately, that way. But yeah, to your point, this image is looking at neurons, but all your cells except red blood cells have mitochondria in your tissues. So this really applies to your whole body. And so there's some out there that think mitochondrial dysfunction is like the cornerstone of health or disease. And so, yeah, you've all made it to this point with me. So you've seen what videos do I send you on the ROF visit? The first one is energy systems, right? If we don't address energy systems and there's an issue there, then we're chasing our tail with higher up the pipeline things. And so we want to look at oxygen and iron. We want to look at glucose. We want to look at thyroid, because thyroid sets the metabolic rate, right? So yes, mitochondria have to be considered. And to Jen's point, many people who walk in are complaining of some level of fatigue. Well, mitochondria might be part of that fatigue picture.